Okay, so now we're getting to the second half, repolarization. So when the membrane potential reaches about positive 35 to 40, I say that because it says 40 on this graph, sometimes it says 35, but it's around that same general value. So the voltage-gated sodium channels become inactivated immediately, and they stop further depolarization. So as the membrane potential approaches this positive value, the voltage-gated potassium channels start to slowly open. So as you're getting closer to that top peak, the voltage-gated potassium channels are going to slowly open. And once you get to the top and the sodium channels close and the voltage-gated potassium channels open and they're completely open, this rapid repolarization happens. So again, if we think about it, intuitively, the potassium channels are opening and there's a high amount of potassium inside the cell, so it is going to rush out of the cell. So we're losing positive charges. Positive charges are leaving the cell, so that is going to make the cell more negative. It's going to bring the membrane potential down. So, and that's why we see this downstroke on the repolarization part of the action potential. Right. So the potassium channels remain open as the cell repolarizes back to its resting potential. So they're open this entire time. And the entire time on the downstroke, the voltage-gated sodium channels are inactivated, which is slightly different than closed, and we're going to talk about that in just a minute. The potassium channels remain open, the cell overshoots its membrane potential, and the potassium channels close at about negative 90 millivolts. So they actually don't close until you pass the membrane potential. So the potassium channels remain open until about negative 90 millivolts. They close, and then at that point, all of those channels, the voltage-gated potassium channels and sodium channels they're not open for business. So we return back to the sodium potassium pump and the potassium leak channels. So they return from that negative 90 millivolts, they return the cell back to equilibrium right up to that 70 millivolts. So the membrane is unable to generate another action potential during this refractory period until the sodium potassium pump and potassium leak channels restore the resting membrane potential and the voltage-gated sodium channels switch from inactive to closed. So when you're talking about voltage-gated ion channels, there are basically three forms that they can take. So there's open, closed, and inactive. When they are open, obviously is the easiest. They're open. Ions are flowing down their concentration gradient freely. When they are closed, Nothing is flowing through, but they are ready to open. So the difference between inactive and open is that when a voltage-gated ion channel is inactive, it is impossible to open it until it switches back to the closed phase. So this is why action potentials have refractory periods. When that voltage-gated sodium channel closes right at the top of the action potential, it is inactive for the entire downstroke and for a little bit of this overshoot while it's repolarizing back to the resting state. So somewhere along right after hyperpolarization, as it's climbing back up to the resting state, it switches from the voltage switches it from inactive to closed, and then it's able to start the process again. But another action potential cannot be generated until that voltage-gated ion channel switches from inactive to closed. So Keith said, are the potassium ions already inside of the cell? Yeah, they should be. Potassium ions, the concentration is always highest of potassium ions inside the cell. So this repolarization phase, the reason why that works so well is because there's so much potassium inside the cell. So as soon as that ion channel opens, the potassium ions are going to rush out. Right, just as Janelle said. They're going to rush out, which causes the repolarization and the return to the resting state. So it releases a bunch of potassium. It hyperpolarizes to about negative 90. And then the sodium potassium pumps and the potassium leak channels slowly help restore it back to the resting state. And the process can repeat. Okay, anyone have any other questions on that?
Where on the graph?
You have a question, Opal?
Is the refractory?
Right. So technically, I guess you can consider the refractory period anytime after the sodium channel opens up, because the real limiting factor on the end of the refractory period is the voltage-gated sodium channel needs to switch from inactive to closed, from the inactive phase to the closed phase. And that happens that lowest point, right where it says hyperpolarization. Between that point and the resting state is when the voltage-gated sodium channels will assume the closed position and they're ready for another action potential. So technically, I mean, I didn't put it on there because I don't think it's super important, but I guess it is worth mentioning. There are two types of refractory periods. There's the relative refractory period and the absolute refractory period. So the absolute refractory period is the one that we're talking about, where it's impossible to generate an action potential because we don't have use of the voltage-gated sodium channels. But once that opens up, as I said, it opens up before we actually get to that -70. So technically, as long as that is able to swing open at the right threshold, we can generate another action potential before we get back to the resting state. It just takes more input because we're not starting from -70 to get to -55, where we could be starting at -80 or -78. So it's going to take a little more input to reach that threshold. Was that helpful? Was that confusing? So the absolute refractory period, it is impossible to generate another action potential just because the sodium-gated ion channels are inactive. There's a quick period where the sodium-gated ion channels become active again, but we are not yet at the -70 resting state. So that small period is the relative refractory period, where it's possible to generate another action potential. It just takes a little bit more energy, a little bit more input. Okay, and now we'll talk a little bit about saltatory conduction. So saltatory comes from the word for jumping. If anybody speaks Italian, speaks Spanish, I'm sure that Latin root looks familiar. And it describes the way that the action potential propagates down the axon. So action potentials are generated at the nodes of Ranvier between these sections of the myelin sheath. So if you look down at this graphic, it almost looks like these little bubbles of myelin that are going down the axon with little arrows hopping at those little points that are not myelinated. So those little points that are not myelinated are the nodes of Ranvier. So basically, it's pretty self-explanatory. I mean, saltatory conduction, it's just the charge is jumping from node to node to node. It's just more efficient that way. It uses less ATP because we're not continuously going down the axon trying to repolarize and depolarize and repolarize every single point. So it just jumps from node to node, and it actually speeds up the process and it makes it more energy efficient. And it also insulates from any background noise, any unwanted input from surrounding neurons as well. So myelin is a mixture of proteins and phospholipids that insulate neurons and speeds up the transfer of action potentials down the axon. It's produced by the oligodendrocytes in the central nervous system and by Schwann cells in the peripheral nervous system. Does anybody have any questions about that? It's just the basic method of how neurons pass information from the cell body to the synaptic terminal. Awesome. So myelin generally makes the conduction faster. I'm not too certain about like the structure, the function, or like the practicality of unmyelinated neurons. I haven't really studied them super in depth, but what you need to know about myelin is pretty much all you need to know is on this slide. It's just a mixture of proteins and phospholipids, and it insulates the neuron. So if you think about like a wire coming out of the back of your TV, there's copper, there's metal on the inside, but it's wrapped, it's insulated because it helps, it blocks out any interference from outside, and it helps the message get where it needs to be quickly. So you can kind of think of it that way. The only difference, of course, is that for a neuron, we have these little nodes of Ranvier, and it causes saltatory conduction. So you can think of it actually like a jumping action. So an action potential starts right over here, right by the body. Once that fires, it is enough to depolarize the membrane all through the myelin sheath, and then it goes to this one, then it goes to the next one, and then it goes to the next one, and then it goes to the next one, and that's just the mechanism of transmitting.
Is this what's going on in muscular dystrophy, where they're not able to respond? Is that
So That might be the case. I know that there are certain conditions where your motor functioning is affected because of a lack of myelin. So I'm
Okay.
not sure specifically if that is for muscular dystrophy, but that is definitely a consequence of unmyelinated axons. Yes.
Yeah, I think those are the questions I usually come across. Yeah,
Yep. Yeah, they like to
fair
relate it
enough.
to
Thank you.
technical, obviously. No problem.